Well, I'm Cesca McInnery. I'm housed here in the Mawson because, in fact, I'm a geologist by training. But I really enjoyed Michelle's talk, and I feel very empowered. And what I didn't know was that I was coming to a self-help session. So it turns out that I am a botanist. You can take a photo and Twitter it if you'd like. Tweet it. That's how hip I am. Twitter it. And so I'm pleased to be talking to you today about the kind of interdisciplinary work that we're doing in my group because, in fact, I guess I am a botanist. I'm going to hold on to this piece of paper and remind myself that. And I'm also part of the Sprigg Geobiology Center, which is something that's really trying to bridge the different fields of geology and biology, which aren't that different, it turns out. I have many co-authors and students I want to acknowledge, only some of which are listed here. And so to tell you about what our group is doing, I thought I would take you on a little trip, both through time and space. And I thought I would start by transporting us, sitting here in the Mawson, to the faraway, beautiful, lush landscape of the Adelaide Botanic Garden across the street, where students of mine and I have been collecting plants. And for what reason do we collect plants? Well, we collect them because we would like to understand the faraway, distant landscape of the Bighorn Basin, Wyoming. Well, you say plants. Why do you need plants? There aren't any plants there to look at. Well, it's not the plants we're interested in. It's the sediments. So those red stripes crossing the landscape there are ancient soil horizons from about 56 million years ago, which, as Bob Hill just told us, was a very different time. And so we're interested in these sediments because we want to understand past ecosystems for their own use, but also to understand how different climates impact those ecosystems. So if we cast our mind back through time travel 56 million years ago, we can see that the Bighorn Basin might have looked something more like this. So it was a very different place, and the way we've reconstructed this is through a combination of looking at fossils, animals and plants, but also looking at the chemical signatures of those animals, plants, and sediments. And the reason that we want to look at this time is because the world was changing rapidly, and that was because of a massive release of CO2 to the atmosphere. Organic carbon was released from somewhere, the source still heavily debated, and that CO2 led to greenhouse warming. That greenhouse warming changed the hydrologic cycle, it changed ecosystems, it changed nutrient cycles, changed soil processes, and we want to understand all of those changes so that we can think about something you probably already have anticipated, future climate change, right? So we have a CO2-driven climate change in the past that operated on a global scale over a long time so that we can look at what might happen in the future. And it was geologically rapid, but it took probably thousands of years to occur. So that is one of the differences, but it's a time that we can use to try to think about the future. So in essence, what my group is doing is using plant chemistry to try to understand these types of ecosystem interactions with climate. And so we look at modern plants because we need to understand how these chemical signatures are set. Then we apply our understanding of those modern plants to the ancient using either fossils or sediments or molecules, as I'll show you in a minute. And the ultimate goal, of course, is to try to understand something about the future and anticipate what might be coming our way. So what chemical signatures am I talking about? Well, one of the main things we like to look at are components of leaf waxes. The cuticle of a leaf contains many different compounds, but some of them are particularly simple, these long-chain hydrocarbons, and they're particularly resistant to degradation, so they stick around for millions of years. These components of these leaf waxes come in different chain lengths, and these different chain lengths can tell us something about the original plant that produced them. In particular, one thing that's very useful is that only plants make these long-chain N-alkanes, as they're called, these particular hydrocarbons. So when we find them, they serve as molecular fossils. So we're not looking at their shape so much as their chemical signature, and in that way, their chemical signature tells us what source they came from. So they're source-specific, and then, as I mentioned, they can stick around for even hundreds of millions of years in the right setting. So we can use them as these fossils. They're basically original chemical constituents of plants that are preserved for millions of years. And when we look at them in the fossil record, we find that, in fact, the relative abundance of these different chain lengths can vary widely. So across that global warming event I mentioned 56 million years ago, we find that overall the chain length, so the relative abundance of these different chain lengths becomes longer. There's much more of the longer chain lengths during the global warming event than before and after. And it's been heavily debated what might be causing this change. There's massive plant community change at this time. We know that in Wyoming, for example, we went from a mixed angiosperm gymnosperm forest to a flora-only angiosperms with many legumes. As you can see, that's about as far as my botanical specificity goes. But it was a very different forest, right? So my botanical colleagues tell me how incredibly different it was. And so we can attribute it potentially to that change in flora, but also the climate changed. And we have an idea that these waxes are important in controlling water loss through the cuticle. So the length of those wax chain lengths can potentially be linked, in theory, to how impermeable that wax layer is. So longer chain lengths should make a more rigid framework for that wax and prevent the water loss through the cuticle. So in theory, it could be that the climate is directly driving what kind of chemical signatures these plants are making. And the only way to really get at this is to look within a species. There's been some work we've done looking across different plant types and asking whether the average chain length, as we call it, the relative abundance of these chains varies. We don't see it changing among different plant types. So perhaps we can find out if it's changing within a species and ask and prove that there's a direct relationship between climate and chain length in at least some situations. And so this is what I'm going to talk about in my first example of two of work coming out of my group. And the thing we needed to do in order to demonstrate that these waxes were changing in response to climate was have some other metric to demonstrate that the plants that we're going to look at are also being affected by climate. And so we're going to bring in another chemical tool here, which is the carbon-isotope ratios of the plant tissues. And these are also known to be responsive to climate because in a leaf there's a trade-off between water loss and carbon uptake. And so when a plant is water stressed, it has to close its stomata and it has less CO2 diffusing in. That leads to more positive delta C13 values because the plants can't be as choosy about what type of carbon, carbon 12 or carbon 13 that they use. And this is true for C3 plants, which is what we're going to be looking at today. So what we've done then is say, well, let's find a C3 species, sample it across a wide range, and use carbon isotopes to tell us that the environment is affecting the plants, and then ask, do waxes change along this gradient? So this sort of chemical ecology project we've conducted on Dodonaea viscosa. This is the honors work of Elise Bunny, who's sitting just there. And this was a really interesting project in which we had, thanks to many collaborators, all sorts of Dodonaea viscosa from across a really wide range of aridity environments. So this is the aridity index shown here. Many plants from across this wide range where we could measure both the carbon isotope ratio and the average chain length of the leaf waxes. And we expected that as aridity increased, we would have an increase in carbon isotope values. And if our ideas about how chain length worked were correct, that we would also see an increase in the average chain length because of that extra impermeability we think is conferred by long chain length. So what did she find? Ta-da! The carbon isotopes, in fact, do become more positive with more arid conditions, so that's confirming that these plants do have a degree of water stress over this environmental gradient. And the average chain length also increases over this same gradient, suggesting that the leaf waxes themselves are, within a species, responding to environment. So this is a piece of the puzzle for why we think the average chain length might have changed when the globe warmed and the environment changed so much during the PETM. And it is sort of suggestive that the plants could be directly responding to that. And if that were true, then the average chain length from sediments, which is a mixture of many different plants, could potentially tell us about past climate. So this is developing this as a paleoclimate tool. Of course, from a biological standpoint, you might ask, well, is this variation plastic? Are the plants just responding, can change as they want? Or is this something fixed in these different populations epigenetically or genetically? So we're now engaged in sort of looking at that in the Common Garden experiment that's going on actually just over our heads. That's run by a whole bunch of people who are interested in the Dodonaea viscosa, Martin Breed leading up that. Collected from